Back to our big picture. This time we are going to discuss this part, and this is adaptive immunity. It's divided into the features. Next, we discuss about the lymphoid organs and tissues, the B and T lymphocytes. We discuss about cell-mediated immunity, and we discuss about humoral immunity. But before we discuss all these parts, we need to discuss or differentiate between innate and adaptive immunity. When an animal encounters an antigen, the light blue curve represents how the innate immune response remains unchanged each time an antigen is encountered. Its activity will be the same for its primary and secondary responses. The dark blue curve shows the primary and secondary responses of the adaptive immunity. It produces a primary antibody response of low magnitude and short duration, peaking at about 10 to 20 days, but during repeated exposure to the same antigen, it results in a secondary response with greater magnitude that peaks earlier. We can therefore have several ways of comparing and contrasting the innate and adaptive immunity. The innate immunity response takes minutes to hours, while adaptive immunity takes days. The innate immunity is limited and fixed, while adaptive immunity is more diverse and specific. Response to repeated infection will be more rapid and effective for adaptive immune response, while it will be the same each time for innate immune response. Aside from comparing these two systems in terms of response time, specificity, and memory, they can also be compared as two immune components. The component of the innate response we discussed in the previous slides are the natural barriers, the different leukocytes, the soluble molecules like cytokines and complements, and the pattern recognition molecules or the PRRs. In the adaptive immune response, we have T and B lymphocytes as the main leukocytes and the secreted molecules in the form of antibodies, although they also secrete cytokines. Lymphocytes are responsible for features of specificity and memory and adaptive immune response. The specificity is due to antigen recognition molecules. These are the specific receptors on T and B lymphocytes, the major histocompatibility complex or the MHC proteins. These are recognition molecules on cell surfaces. And thirdly, you have your antibodies, which are the secreted form of B cell receptors, synthesized before these B cells are exposed to antigens. The next feature, which is diversity, is based on a huge variety of antigen receptor configurations, essentially one receptor for each different antigen that might be encountered. This is mainly achieved by variable recombination of gene segments prior to exposure to antigen. The recognition of an antigen by the lymphocyte with specific receptor occurs mainly in the local lymph node, and this induces the activation, proliferation, and differentiation of the responsive cell, and this is known as clonal selection. Only the clone of this lymphocyte that has recognized the antigen proliferates and generates progenitor cells. The clones of these lymphocytes become plasma cells in the case of B cells and effector cells in the case of T cells. Memory cells are both descendants in the expanded B cell and T cell clones. When the antigen is encountered again, these cells mimic the reactive specificity of the original lymphocytes that responded to the antigen, and this is the basis for the fourth feature, which is immunological memory. As was mentioned in your RBC physiology lecture, during early fetal development, blood cells are produced in the mesenchyme of the yolk sac. As the fetus develops, this function is taken over by the liver and the spleen, but during the last months, the bone marrow becomes the dominant organ for hemopoiesis. As far as immune cells are concerned, we have the bone marrow and the thymus as the primary lymphoid organs. These are also called generative or central lymphoid organs because these are the sites where the lymphocytes are provided with growth factors and other molecular signals needed for lymphocyte maturation. The secondary or peripheral organs include these lymph nodes and spleen. They are encapsulated, and we also have unencapsulated secondary lymphoid organs, and these are divided into NOT or your nasal associated lymphoid tissue in the tonsils and adenoids. You have BOLT, the bronchus associated lymphoid tissues. You also have SALT, the skin associated lymphoid tissue in the axillary lymph nodes. A GUT or the gut associated lymphoid tissues are present in the mesenteric lymph nodes. You also have them in the Peyer's patches. You also have this reproductive tract associated lymphoid tissues that include your inguinal lymph nodes, and not shown here are the para-aortic lymph nodes. Collectively, they are known as the MALT or the mucosa associated lymphoid tissues. B lymphocytes mature partially in the bone marrow, enter the circulation, and migrate to the spleen where they complete their maturation and then populate secondary lymphoid organs. The T cells mature in the lymph node, in the thymus, then enter the circulation and migrate to secondary lymphoid organs. After birth, some lymphocytes are formed in the bone marrow. However, most are formed in the lymph nodes, thymus, and spleen from precursor cells that originally came from the bone marrow. We have here the spleen. It is a highly vascularized organ, and its major function is to remove aging and damaged blood cells and particles from the circulation, and also to initiate adaptive immune responses to blood-borne antigens. It's located in the left upper quadrant of the abdomen. The parenchyma is divided into your red pulp and your white pulp. The white pulp is lymphocyte rich. Okay, and we have this T cell zone called your pulse or your periarteriolar lymphoid sheath, and then we have this follicles, which are your B cell zones. Shown here are the lymph vessels and the lymph nodes. Lymph nodes are encapsulated and are scattered throughout the body. There are cervical lymph nodes, axillary nodes, inguinal nodes, and these are palpated during clinical examination if you suspect that a patient is having an ongoing infection in these specific areas. Antigens are carried from the tissues to these lymph nodes by the lymphatics. This is where initiation of the adaptive immune response occurs. Within the organ are B cells, specifically in these areas called the B cell zone. These are the follicles. And T cells reside in the paracortex. The captured antigens carried by the lymphatics can be seen or recognized by these B cells and T cells. Please remember these parts because we will be mentioning them again later. Shown here are the microscopic appearance of B cell follicles with the germinal centers. You also see the T cell zone, referred to as the paracortex. These are the two arms of the adaptive immunity. So you have your humoral immunity and your cell-mediated immunity. Yeah, the humoral immunity will be fighting against extracellular microbes, while cell-mediated immunity, or your cell-mediated immune response, will be against your phagocytosed microbes in macrophages and intracellular microbes. The B lymphocytes will be mediating your humoral immunity. They will be secreting antibodies. While in your cell-mediated immunity, you have your helper T cells and your cytotoxic T cells. Your helper T cells will activate macrophages. Your T cells will kill infected cells and eliminate reservoir of infection. Now, your antibodies will also block infections and eliminate extracellular microbes. We will see the details of this. Big picture again. This time, we focus on the primary lymphoid organs, the bone marrow and the thymus, because we will be discussing the lymphocytes. So here we have a picture of the thymocytes in the cortex of the thymus. Thymocytes are the baby T cells found in your thymus. Let's now look at the development of T cells. The thymus is an organ surrounded by a fibrous capsule that encloses multiple lobes, each of which is separated into two major regions, the outer cortex and the inner medulla. Each stage of development occurs in specific areas of the thymus, and the thymocytes enter the thymus via blood vessels that are in the corticomedullary junction. And then they travel into the cortex, proliferating first in the subcapsular cortex. As they mature, they migrate from the cortex into the medulla and ultimately exit via vessels in the corticomedullary junction. Two phases of T cell development are early thymocyte development and T cell receptor interaction. In the early thymocyte development, there's commitment of hematopoietic precursors to the T cell lineage. There is also initiation of the T cell receptor gene rearrangements and expansion of cells that have undergone these gene rearrangements successfully. The second phase of T cell development is T cell receptor interactions. This involves positive selection and negative selection. In the positive selection, T cells not binding to self-MHC will undergo apoptosis, while T cells binding to MHC are selected. In the negative selection, T cells binding strongly to MHC and self-antigens will undergo apoptosis, while T cells binding weakly to MHC will be selected. Mature T cells express either CD4 or CD8 with a single positive and leave the thymus with the potential to initiate an immune response. Now, why are these phases necessary? They are necessary because of the following reasons. First, T cells should have an array of receptor specificities. Next, TCR should recognize only peptide antigens combined with self-MHC molecules, and T cells should be largely tolerant to self-MHC peptide complexes, which means to say that they should not initiate a response with them. This is called self-tolerance. B cell development begins with a hematopoietic stem cell and passes through several stages until it reaches the pro-B cell stage. At this stage, the precursor cell is irreversibly committed to become a B cell, and the antibody gene rearrangements begin. In some, almost unlimited diversity is created from a limited number of V gene heavy chain rearrangements. The first antibody produced by an immature B cell is of the IgM class. Once the completed immunoglobulin is expressed on the surface of the cells, the immature B cell, now a transitional B cell, leaves the bone marrow to complete its maturation in the spleen. Mature cells acquire surface IgD in addition to IgM, and they migrate to the secondary lymphoid organs and tissues where they respond to foreign antigens by proliferation and development into memory and plasma cells. These are the stages in the life history of lymphocytes. So in the generative or primary lymphoid organs, there's the development of the B cells and the T cells. B cells remain in the bone marrow and continue their development in the spleen, while T cells have their maturation in the thymus. The products of this development are called naive lymphocytes because they haven't encountered antigens yet. Activated lymphocytes undergo proliferation and differentiation to produce effector lymphocytes. Effectors of B cells are known as plasma cells, and they produce antibodies. Effector cells of T lymphocytes are the helper T cells and the cytotoxic T cells. They produce cytokines. Both have memory lymphocytes. Once mature, naive B and T cells enter the blood and distribute themselves throughout the body. After a brief transit time of around 30 minutes in the blood, nearly 45%, around 40% of the mature naive lymphocytes produced by the thymus and bone marrow travel by blood directly to the spleen. So that's around 50%. They stay there for approximately 5 hours, and an almost equal proportion of lymphocytes enter various lymph nodes, around 40%, where they remain scanning the environment for about 12 to 14 hours or 12 to 18 hours. Now, a small number of lymphocytes migrate to the secondary lymphoid tissues in the skin and gastrointestinal and pulmonary and genitourinary mucosa. Several protein interactions regulate lymphocyte homing to specific tissues. These are actually controlled by your chemokines. There is continual lymphocyte recirculation, and this recirculation maximizes the number of naive lymphocytes that encounter antigen. So an individual lymphocyte may make a complete circuit from the blood to the tissues and lymph nodes and back again once or twice a day. So that's a complete circuit. B and T lymphocytes travel into the lymph node from the blood. They migrate into the paracortex across high endothelial venules. So the T lymphocytes browse the surfaces of cells on this area. If they do not find antigens here within 16 to 20 hours, they leave the lymph node via this efferent lymphatics in the medulla. Now, B lymphocytes travel into the follicles here, and they browse surfaces of follicular dendritic cells. Now, if they do not find antigen, they will also leave the node via this lymphatic, efferent lymphatics. Now, dendritic cells and other antigen-presenting cells migrate into the lymph node through this afferent lymphatics. They join the resident dendritic cells in this T zone here, where they are scanned by lymphocytes. Antigens typically arrive at the lymph node via afferent lymphatics also, entering the subcapsular sinuses. The antigens are either unprocessed or processed peptide presented by antigen-presenting cells. So T and B cells are directed to their respective zones by chemokine receptor chemokine interactions. Now we have here a table showing the comparison of your B and T cells. As we have discussed, the site of maturation of the thymus of the T cells is in the thymus, the B cells in the bone marrow, and the main location in the lymph nodes of your T cells is in the paracortical area. The B cells are mainly in the follicles. Now, both have memory cells. The T cells will protect us against intracellular microbes, and they also provide help for antibody responses. Now, the B cells will protect us against extracellular microbes. And these are their products. They have cytokines as their products, the T cells. The B cells will have antibodies as their products. The T cells will require MHC for recognition, while the B cells will not require MHC for recognition of antigens. These are your mature lymphocytes. Only 5% of these are found in circulation, where they constitute around 20 to 35% of all WBCs. In letter A, you have a small lymphocyte. This is a typical resting lymphocyte with a thin rim of cytoplasm. In B, you have an electron micrograph of a lymphocyte with indented nucleus. Okay, so letter D, in letter D, you have plasma cells. The nucleus is eccentric. The cytoplasm is strongly basophilic due to the high RNA content. Now, letter E, okay, letter E is a plasma cell. And you can see that the endoplasmic reticulum is very prominent, and this is associated with synthesis and secretion of immunoglobulins. Let's now compare the lymphocytes with other WBCs as seen in normal WBC count. So this is one of the routine blood works ordered in the clinics. We order CBC and differential count of your white blood cells separately. Lymphocytes enter the bloodstream for the most part via the lymphatics. And at any given time, only about 2% of the body lymphocytes are in the peripheral blood. Most of them are in the lymphoid organs. So the most numerous white blood cell will be your neutrophils. If there is increased neutrophil, that may indicate acute infection, acute bacterial infection. If you see increased eosinophils, that may indicate increased parasitic infestation. okay, or allergies. The increased basophils will also indicate that. And when there is increased lymphocytes, there may be increased viral infection.